Yeah, it was this
There
morning.
it is.
Hello, how are you?
Hello.
Hi.
Gee, finally you're late.
Yeah.
Oh,
Well, welcome to the meeting. We have a big announcement this week was the Google Summer of Code student announcements. So we got 4 in this group. And yeah, so we had the 2 people who applied to do the digital microspheres and the 2 people who applied to do the GNNs.
Okay, got to get a whole bunch more pictures.
Yeah.
Yeah. I was thinking of like those BB guns and getting a little one of those and then just putting a whole bunch of marker dots on it.
Okay.
painting a 2mm sphere is not easy.
Yeah.
No, I was going to glue it down to the cover slide and then carefully dot it with a whole bunch of different felt markers.
Okay.
Good luck.
Okay. Good luck, yes.
Yeah.
Okay.
Yeah. You mean to get some sort of like baseline or benchmark data for the scope, or I wasn't sure what you were referring to. Why would you do that?
In this microscope, you can see the thing behind me,
Yeah.
and it's geared right now for peppercorn, sort of, although there a couple of the top microscopes aren't.
Bradley,
Yeah.
12 years ago, I bought a software for making an outline model of the world.
Okay.
And then I was at MIT visiting there, and I had them print it onto a 2mm diameter sphere.
Okay.
Okay? And it didn't work at all.
Oh.
It was an absolute mess.
Yeah.
They didn't have the resolution.
Oh,
I don't know
right.
if they have it now, but
Yeah.
so getting a nice set of coordinates onto a sphere is not going to be trivial.
Right. Right. Yeah, it's usually yeah. So that's we'll think about how to do that. So I guess I wanted to go through each person, and doesn't look like Jihang is here right now, but the 3 people who are here, if you could introduce yourselves and maybe talk a little bit about your project and what you hope to accomplish this session or this summer. So I think Karan has to go. If he could go first.
You have to wake them up first.
Oh yeah. Hello?
Try chat.
Okay, yeah, I'll try. I mean, if Karan's not ready, we can go to someone else, but I just wanted to make sure everyone got a chance to do that. Okay, he'll introduce himself in the chat.
Oh, there he is.
Yeah. So I'll read the chat when he
Hello.
types.
Hello. Am I audible?
Oh, there you are. Okay.
Hi,
Yeah.
hi, Bradley.
Hi.
Hi, Richard. Hi, Susan.
No, you have
Hi,
anything
Harikrishna.
next.
Yeah. So, introduction-wise, I'm currently doing my bachelor's degree from Amrita University. My core subjects are electrical and computer engineering. Like we have computer architecture. It's like a wide array of subjects where we get into the hardware abstraction layers to the software side as well. So apart from that, introduction. Okay, wait. I think there's a lot of disturbance coming from the background.
He's trying to find a quiet
Yeah,
place.
yeah,
There we
I'm
go.
back.
Okay.
Yeah. So the approach related to the digital microsphere, like I had given 2-3 presentations before. So the idea is pretty much still the similar idea. I'll be extracting each embryo image from the sample data set, and from that I'll be okay. I'll just write it down in the chat.
Okay.
That's more convenient because
Artie's going to write it in the chat, so I'm going to read what he says. Yeah, so he's presented on this a couple times before, and we've been working on this project a little bit, getting a sense of how to approach the project. I think that was a smart move and I know Harikrishna has done the same, just to get a sense of how to do it. I don't think we have everything worked out, but I think one of the things I'd like to do is to be complementary here, and Dick has agreed on this, where Harikrishna and Karan work together when you can and make sure that you're not overlapping. So you don't do the same work twice, basically.
Right.
Yeah.
Overlapping is the goal.
yeah. Well, yeah, we want something at the end that is, but they're different.
Images.
Right. There are a lot of different things that can be sort of not reproduced. Okay. Well, Karan is typing that out. Harikrishna, would you like to go and introduce yourself?
Hello.
Hello. Yeah.
Yeah. So, well, currently I am studying a BTech from Amrita University, which I'm doing BTech in artificial intelligence. So till now I'm about to complete my first year, and so related to the topic, this topic. My goal this summer will be to create a computational tool which will basically map the 2D images of the embryo onto a sphere, and also have to take care of the overlappings. So yeah, till now I think I've made a good progress, and by contributing with Karan, I think it will be eventually done.
Okay, yeah. Yeah, that's good. Thanks for that. Let's see if I can pull up. Okay. Karan still hasn't gotten in the description. But yeah, so yeah, I've seen some of your work that you've done. You've done some pretty good work so far. And again, we'll have to try to figure out how to not overlap in places where we don't need to. So that's good. And then Wataru. Hello.
Hello.
Hi. Go ahead and introduce yourself.
Yeah, thank you. I'm a 50-year student at Kyoto University, and I am currently pursuing a medical doctor. And I belong to a laboratory in the Graduate School of Informatics in Kyoto University, and I am developing methods for visual reconstruction of what is seen by subjects by inferring the features of the hidden layer of DNNs from fMRI data, functional MRI. Yeah, and I will develop in this summer. I will make some GNNs that resemble actual biological networks found throughout development, but I have decided the details, so I have to start with searching for some interesting theory or something.
Okay. Yeah. Yeah, that sounds good. I want to, in the next couple weeks, I want to highlight some data sets and things that you can use to start off with. I know we have some, but I want to be able to bring them to both you and Gihan's attention. So, it's going to be a matter of kind of figuring out, what data set you can work with to extract data from. And we'll go over that a little bit in the following weeks. So we have data that, we use a lot of secondary data, a lot of microscopy data. So the Digital Microspheres project is using, a certain data set. And you may or may not find that useful. You may find other types of data useful. In the past, we've used a lot of data from C. elegans development. So this is where we're looking at cells and defining cells in their space, and then extracting those data through segmentation or other types of techniques. So, this is something that we have a lot of secondary data sets sort of marked that are, we know that are good to work with. They're sort of good benchmarks for this sort of thing. And then that might help you, kind of get into the data and start working out, something concrete in terms of, how you might build these networks or these embeddings. Again, like with Jiahong, I know that I don't know if you two have talked yet, but I want to make sure that, again, you don't overlap in key areas. So I don't think Jiahong has proposed anything like really close to what you've proposed, although you didn't give the full details. So that's something we'll have to work out. I just want to make sure that you don't do the same thing, exactly the same thing, and that's something maybe you could work on different parts of this or different data sets. I want to talk to him more about that before we start getting into really deeply into this. But welcome aboard to the group here.
Oh, Bradley, can I ask a general question?
Yeah.
So the question to everybody. If you have access to a 3D printer, could you please let us know the maximum resolution of it?
Okay.
I have a 3D printer, but I don't know its resolution at all.
Oh, okay. Go find out. And we can see if we can do 2 millimeter spheres.
2 centimeter. I don't know.
No, not 2 centimeter, 2 millimeter.
Yeah. Okay. I need that glass printer that I was talking about. I don't know.
Maybe. I don't care what material, as long as you can visualize.
Well, there's a 3D glass printer. It sounds really interesting, but...
Yes, but I don't think you want the light to go through your object. Well, it won't imitate an embryo, an axolotl.
That's okay. You could paint the outside.
No, you can't.
Sort of. Thoughts.
Susan, one thing you could consider is if you can get a little glass bead, which would be smooth on the outside, you could try coating it with an organic material and then shrink wrapping it and look at the wrinkles.
Oh, well, you could paint it and then dry it, and then the paint would crack.
Maybe.
I'll try stuff. I think I have some glass beads. I'll take a look, or else I could get one of these BB gun things and melt it a bit. I don't know.
Okay.
Yeah. So you're trying to build a 3D printed model of an embryo, or?
Yeah, I'm just trying to build something that you can do the 3D imaging with.
Okay.
Yeah, it has to be
okay.
three-dimensional, and it has to have texture on the surface, and it has to be 2 millimeters in diameter.
Okay. So to fit into the stage, yeah, I see.
Yeah.
I still haven't been able to people on Kijiji are trying to sell me salamanders for $350. So
Oh,
I'm doing. I don't need it to be a whatever golden thingy that you have. It just needs to be black, wild type, something or other. It doesn't have to be a specialty female axolotl. But anyways, I'll keep trying Kijiji. Sometimes they come up with things.
yeah.
Susan, can you get small iron spheres and let them rust?
Oh,
That might produce a texture.
yeah. I'm just. I don't know if they make ball bearings that small, but I can
Well,
look.
they may not make them of iron. They're probably made of steel, so they won't rust.
Well, you can do things to steel. Take a hammer to it.
Yeah, use
You'll break it.
use an abrasive surface like sandpaper or something to roughen it up a little bit. I don't know.
Yes, yes. I'll
Very
figure
fine
out
thing.
a couple of things. I'm going to try a couple of things. So.
Yes.
Okay, that sounds great. And then yeah, we can get some images.
And then we can publish it in the journal Hard Matter.
Yeah. Instead of soft matter, right?
Sure. No, you need to publish in the journal Soft Matter and give them a bad time.
It's
Yeah.
like,
Okay.
yeah.
Okay, Karan has just given us a long message.
Yeah, so he gave his project description. I have divided the problem into three main subproblems. The first is segmenting the embryo image from the background of the image for all samples. After this, the second part is based on using the embryo image and the contour of that embryo image to create a 3D point cloud by rotating the embryo outlines or contours about the axis of rotation. And then after generating the 3D model from this 3D point cloud, I'll be applying cylindrical projection techniques to project the image onto my 3D generated model. So this part will require stitching all 8 embryo images together into one cohesive equirectangular image that I will then project onto my model. So you're building this, you're taking these images, you're stitching them into this cohesive image, and then you're sort of warping it into the model from the 3D point cloud. I guess that's how it works.
Can I
Yeah.
make an outlander suggestion?
Yeah.
Okay, I once made a 3D point cloud, and I viewed it in two directions about 6 degrees apart. And that made a very nice stereo pair. You could actually see it in 3 dimensions.
Yeah.
Okay. So it might be a useful tool to make a stereo pair and look at these 3D point clouds.
Yeah.
Okay.
Yeah. They have, I think they have tools to like optimize the distance between the images, so the overlap that you need to have a coherent stereo.
Yeah, 6 degrees is about right for vision,
Yeah.
for stereo vision.
Yeah, that would be good. Yeah, I think that sounds like a good project description. So again, this is something that we'll be working on over the course of the summer. And again, we have these two groups, the digital microspheres and the GNNs. And so I want you to, we'll have these meetings once a week, Mondays, and then we'll try to catch up on Slack over the course of the week. So I want people to discuss things between yourselves. I'll try to answer questions. In the next few weeks, I'm going to be giving you these materials in terms of data or, things that you might need to sort of get going. And so I hope, if you have any questions, let me know. Don't be afraid to ask, or you can ask Susan or Dick here about some of especially for the digital microspheres, because this is something that they've been working on, I think, for a while, this idea. So we'll see. Yeah. So the next few weeks for the students is going to be a community period, and that's built into the program. And so check your schedule that they have on the website. They kind of they talk about the community period, which is I think 2 to 3 weeks, maybe even a little bit longer this year, but I'm not sure. And then you start your projects in June, and then you go through. You can go through to September, or go through. I think the last date you can turn things in is November, so it's a little bit flexible with respect to timing. You have 175 hour projects. Both projects are 175 hours. So that means you'd be working for, I think, 12 weeks at maybe 20 to 30 hours a week doing these sorts of things. And you'll get your stipend. But you can extend that time out until, I think, November. So if you have exams or something, you can extend it by a couple weeks. You'll have to consult the schedule to figure out exactly how you want to do that. And then, I made you, in your proposals, I think I told people that I wanted to see a schedule. So like a table where you had week by week activities planned out and, like a deliverable at the end of that week. So the reason I told you to do it that way is A, it keeps you on task. And so it keeps you accountable every week. It tells you what you need to, catch up on and, where you need to go next. But the other thing it does is it allows you to move things around accordingly. So if you miss 1 week because you have exams, you can pick up. You might have to shift things around, but at least you know where you are. So the other thing is, when you do these weekly meetings, you're going to be presenting something at every meeting. So maybe not next week, but definitely when we start the coding period, I want to have a report once a week. Each person will go around and, you can tell us what you've done. Maybe, ideally, I'd like to see like a screen share of some things that you've done. It could be like a notebook, like a collab notebook. It could be like some slides. It could be, some visualization, just something to show that, you're progressing and that, especially if you have questions or if you have a problem that arises, that's a good time to sort of bring that up and we can help you debug things. Maybe not like some really big problem, but just something that, if you're having a problem, you need advice on it. That would be a good time to do that. And I just want to make sure that people do this every week because it's, it just helps you keep on task and it keeps you going. So that's really what we want to do because you don't have a lot of time to, kind of figure out what you want to do. You have to get into this, hit the ground running. So I know Wataru is, he's trying to make sure that he has a grasp on things. And I know Jia had a more detailed plan, but still I want to make sure that, especially for the GNNs project, that we're going in the right direction on that. That project is a little hard to sort of grasp, mainly because, we haven't really done much work on it before. So that's something we'll just have to kind of figure out as we go along. We also want to have like some really nice targets, so we know kind of what we're doing week by week and that we make progress on that. Yeah. 3 steps forward, 2 steps back is progress. So yeah, we'll see. I mean, don't worry about like, if you get stuck on something, and I think I've told people this in the application period. If you get stuck on something, don't spend like 4 weeks trying to figure it out. Ask around. There are ways to find out on like Stack Exchange and things like that about simple problems. But if you're having some more fundamental issue, please let me know because I'll, we can clear it up pretty quickly. I think if we talk amongst ourselves. So that's something we want to make sure we're active about. We don't want to leave things for a couple weeks because we don't really have that kind of time to do that. But yeah, go ahead.
I had a brief, some information about the images. They look like they're ovals. because they're being taken on an angle. So the round peppercorn will probably look slightly oval. And my suggestion is to just stretch it back to being a sphere or circle.
Okay. Yeah.
Because it's round. I mean.
Okay, Susan, that would be using the minor axis as the diameter. You know, if the image is an ellipse, it's major and a minor axis.
Yeah.
So.
Well, it's a combination.
Yeah, but would you want to use the minor axis as the real diameter?
I think it would be a combination.
Oh, really?
I would think so.
Okay.
Well, here's your, now they're oval.
Yes. But
Oh, that's a major axis.
Oh, that's a major. That's a major.
Yes, a major axis. Yeah. Okay.
I see.
All I had to do was get out my thing. Yeah.
Yeah.
Major axis. Major axis.
Okay.
Yeah.
Okay.
Yeah.
Well, that's good. Yeah. Oh, go ahead.
It's okay. That I'll get some more images with that. Like I said, it's set up for 3mm peppercorn. I can surely find some other things to image, and then there could be a variety.
Okay. Yeah, that's good. Yeah. So I also wanted to point. I know Wataru and Karan had to go, but I have some other things. So another thing for the community period is we're going to have. I want people to also get to know the OpenWorm community. So I'll try to facilitate some interactions with the rest of the OpenWorm community. Well, there are a lot. If you're in the Slack, there are a lot of different channels that exist. So it's not just the DevoWorm channel. It's also.
Can we wake up Steve Larson and have him do a very early morning session?
I don't know. He's pretty busy these days, but actually, he's got some talks online. I'll put together a list of things that we have.
Okay.
He's done some talks on OpenWorm, and then that'll be, you can view those and you get a bigger perspective on the OpenWorm community. But there are also other channels that you can, I'd like to encourage people maybe to check out, maybe get into some conversations on. So one of the things that OpenWorm has is a data set on movement of C. elegans. So this is where you have worm movement, and they're using computer vision to segment some of those movements into, distinct sort of identities. So like in C. elegans, they have these stereotype movements that you can classify. They're not ambiguous. They're reproducible from worm to worm, and you can easily see them when you look at a culture. So, they've tried to work out all those movements, and so that's something that maybe is a good source of data for some people. For the GNN people, that might be a place to go and look at some. Maybe it's not really embryos, but it's enough data to sort of benchmark your algorithm and say, oh, this is something that's structured. Maybe we can create an embedding from this. So I want to facilitate some interactions there as well. The other thing I want to do during the community period is, and I'm going to share my screen, talk about open source and sort of engage in some open source, a broader open source education. So I have a set of readings here that I've collected over the years, and these are on different topics in open source. So we have this set of open source books that I have. So I have this book on producing open source software, and this is a nice book on like the production process behind open source software. So this goes through a lot of different things with respect to, if you're creating a program and you're licensing it as an open source product, what goes into that. There's a lot of sort of, there's production, there's keeping your software up to date. So there are a lot of things in there that are, things you have to think about. And this is especially useful if you want to go beyond the summer of code and you want to think about, what do you want to do with your software. In years past, we've done, we've had people be maintainers of software. So, when you create your software at the end of the semester or the end of the summer, it can either sit there and, collect moss, or I guess digital moss, or or you can improve upon it. And so I'm not saying that you have to be burdened with improving it, but there is this aspect of getting people involved to, make commits to the software, and there are other types of things that you can do. You also maybe want to license it or advertise it to people so that you get a user base, so that people start using it. So this gives you a bunch of different tips and tricks to these things. It talks about developing, and it talks about some of these other things. I just want to make sure that people have access to this information, so that they can make the most of their open source experience. We also have some other readings in here on open data, on new types of institutions. And this has to do with like the types of institutions like OpenWorm, and how that's facilitating a lot of this open source software production and other types of activities. So, and then I have some things on licensing, which actually this is something Ryan Merkley used to be on the board of OpenWorm, and he wrote an article on licensing creative work and how attribution is gratitude. So this is an article on open source attribution, which is a little bit different than the way we usually think about open source, or the way we usually think about attribution. So this, it involves, there's an emphasis on sharing, an emphasis on collaboration, instead of, putting things behind a wall of an intellectual property wall. The intellectual property it's managed differently. And so this kind of goes through some of that. I'm going to add to this repository, and I'm going to make it public here during the community period. And I hope people maybe we'll go over next week one or two of these resources, and we'll kind of get an idea of what it means to work open and what it means to build something open source and how we facilitate that. So I just wanted to mention those resources, because I think they're useful. And I'll put them in the Slack as well. Also, I want to make sure that we know that we have a GitHub repository. And I don't know, I sent a message out to the new students, and I mentioned that it was a huge email that had all these different references in it, or resources. So this is something that we want to go through. I have a repository here, GSoC 2022. This repository is 2 sub-repositories, or subdirectories, D-GNNs and Digital Microsphere. And so I want the students to, I'm going to invite them to join these as appropriate. So Harikrishna and Karan will be in Digital Microsphere, and Wataru and Jiahong will be in D-GNNs. And so those 2 places will be where all their work is pushed. So this is labeled by project. So this is, I think, Harikrishna's project. This one is Karan's project. That's Digital Microsphere. And then in D-GNNs, we have DevoGraph, which is Jiahong. And then this is Wataru GNN's developmental networks. I'll send out an email to make this clear, how this works out. And where I expect people to push. It'll basically be, where I'll invite the students into this space as collaborators, and then they'll be able to push their work. And I want to basically have people update their work maybe once a week. They can create a fork of this repository and update, and then push back to this main repository maybe once or twice a week, maybe once every 2 weeks, depending on how much is being done and how much needs to be committed. I basically want to enforce using GitHub as a main place to store code, because I think, we don't want to store code on our local machine, because if the local machine crashes, you lose your work. So be sure to use GitHub for everything. Be sure to push at least your local fork. And then when your fork is ready to be updated, maybe once a week, make it a habit of it. Push once a week and label the pull request as I've done this. And sometimes there won't be anything in the push much at all, maybe a couple files. Sometimes you'll have a lot of stuff. But I just want to get an update through GitHub and to make sure that we're, keeping on track. I think GitHub updates, updated pushes to the main repository, I think are a good way to enforce progress and keeping things moving. And it, it gives you this account of what you've done. So I made some changes to this file, I've added this file, and you can see the progress before your eyes. So that's good. So I'll send out those invites this week. Okay. Any questions about that? I know Harikrishna is the only one here is in GSoC, so but we'll have this available later, and I'll send out.
GitHub never goes down.
Well, it does. It can, but it's the thing about GitHub is you can have a version of it where you can download a desktop version and have everything local, and then have everything up there, in the cloud. So you can, things can be synchronized. When it goes down, it just goes down and it comes back up, and
Okay.
yeah. You were going to say something, Harikrishna?
No, I just said that I don't have any doubts.
Okay, yeah. So yeah, I think this will be good, a good way to do things, and I'm looking forward to starting off, getting going on this. Like I said, this first couple weeks is the community period, so I want to make sure that people are, getting to know their community. And that means, being in the meetings, and you can watch the meetings afterwards, but, having some Slack interactions and just learning a little bit more about OpenWorm. And I know even Harikrishna and Karan have been in the meetings here, but I don't know how much they're familiar with OpenWorm overall. So this is a good opportunity to learn about that. There's a lot going on, or a lot that has gone on in the past, and so it's good to learn about those things. Then again, I don't want this first week or so to be overwhelming, because you're going to get a lot of information, but just kind of keep those emails available to yourself, and then kind of go through them and use them as references for later, because there's going to be a lot of detail you're going to have to absorb to get started. But once you get started, it shouldn't be too bad. I've had in previous years, it hasn't been that bad. I've tried different things in previous years. I've tried to get people to do presentations of their work, at the beginning of the session. And I think I mentioned that in the introductory email, is that I'm kind of wanting people to do a very short presentation. And I think Harikrishna and Karan have kind of done that already. But I want to have I want everyone to kind of get together, maybe about a 10-minute slideshow of what their project's about. And the reason I want to do that is because I want to have a record of, when you start out, what is your project about? And then at the end, you're going to add on to that and say, this is what my project ended up being, and with results. So you can see, you'll be able to see the progress from the beginning to the end. So, this is before and after. You get to see what worked and what didn't, and then you get to see the finished product. I think it'll be useful for people to, kind of for future years to see, and for yourself to see your progress. So, okay. So that's it. Yeah, go ahead.
Yeah, Bradley, on this, Harikrishna and I had some discussion about what doesn't work.
Yeah.
Okay, and I think if you invest some time in something that doesn't work, it's worthwhile when you write it up to include that,
Right.
because that keeps other people from making the same, I would call it a mistake, but the same path that didn't work.
Right.
Okay?
Yeah.
And for example, he tried simulated annealing for the overlap problem, and for some reason it didn't work. And it would be nice to see it work. A write-up, a short write-up, but why it didn't, what happened, and is there any explanation for why it didn't work?
Yeah.
Yeah, sure. I'll write.
Okay.
Yeah. Yeah, that's definitely true. I think what didn't work, what worked, and kind of like, you're making this progress towards. So you probably proposed, I'm going to take the number of, potential approaches, and then you've tried a couple of them. Maybe some worked, some didn't. So that's good, because usually in our projects we usually encourage people to think about like what the potential solution is. We don't hand them the solution and say, here you go. So it's useful to think like, well, to start off by thinking, well, what's most likely to work? And then, it's like no one really knows that answer. You just have to kind of guess from what you've been exposed to. So I might pick something that is a thing that I'm familiar with, and there may be solutions that I have no idea exist, and they might be better fits for it. So that's something you just have to learn by trial and error. But it's also good to know kind of what people have done in the past, because it's like, okay, at least I know now this new method, the pros and cons and so forth. Okay, so let me think where we want to go next. I think I'm going to go to some of the stuff that we have. I have some things in the presentation bin or in the paper bin that I want to talk about a little bit. There's been this discussion or a couple of papers that have been come out recently on the future of developmental biology. So there's this idea that like developmental biology is all washed up, that there's not much more to discover. And then other people think, not my words. And then there are other people who think, well, there's a vast frontier of things to talk about. So, there have been a couple of papers that have come out. This is one of them. This is Developmental Biology is Poised to Discover Altogether New Principles in Biology in the 21st Century. And this was published in Developmental Biology this year. So this is something that is just kind of a sort of a recent conversation, but it's something people were talking about. So the abstract reads, in the 20th century, developmental biology spearheaded a revolution in our understanding of complex biological problems. Its success rests in great part on a truly unique approach that has recruited a diversity of systems and research organisms rather than focusing on isolated cells and molecules. So a lot of biology has been very much focused on specific genes or specific proteins or, specific cells or cell populations. If you think of neuroscience, you don't think in terms of model organisms so much. You think in terms of circuits, or you think in terms of different types. In insects, they have different types of neurons that do specific things. So people study those just as like a topic. So these are, this has been the focus. In developmental biology, by contrast, you think about like different model organisms, like zebrafish or like axolotl. That's why we talk about these different species, because those are the comparators. Like you look at the process of development in those model organisms. Now that's not to say that there hasn't been work on, say, like the molecules of development or the cells of development. There have been, specific cells that are, or cell populations that are interesting in development. But they're kind of making this distinction between the two approaches. And then, developmental biology has also employed a wide variety of technological and intellectual approaches. But what will developmental biology contribute to this century? Advances in technology and instrumentation are presently moving at neck-breaking, I guess they mean breakneck speed, and herald the advent of an age of technological wonders, in which previously inaccessible biology is now tangibly within our grasp. So they give the example of single-cell RNA-seq. So this is a technology, of course, that you can take a single cell, you can sequence the RNA in the cell, and you can map it to a genome sequence. So, we have genome sequences about what's in the genome, what the genes are, what the regulatory elements are, and you take these little fragments of RNA that you find in the cell, and you can sequence those at maybe 100 base pairs, and then you can match them up to the source in the genome. So there's a lot of computational work involved in that, but basically it gives you some candidate regions where that transcript came from, and you can say things about its function and things like that. So this is a nice technique, and having in a single cell, of course, in development is good because, in the case of C. elegans, a lot of the cells are very unique in terms of their function. So you have a cell that's, a cell that is born of like a precursor cell. They divide and they differentiate into their adult form, and they retain their sort of identity as a single functional type throughout development. It's different than most other parts of development. But having the single cell approach in C. elegans is nice because you can see what's being expressed as the cell goes through its divisions and then it becomes differentiated. So that's some, but of course, single cell RNA-seq is problematic in some ways because it's very hard to understand what the data mean. So, what does it mean to have a bunch of genes upregulated, as they say, meaning that there are more copies of the RNA? Or what does it mean to have the genes downregulated, which means there are fewer copies of the RNA? Well, it's hard to know exactly what that means. It may mean that it's, there's some functional thing going on, or it could just be some fluctuation. And this is, these are things that we think about in the 21st century. But so this technology, single cell RNA-seq, has revealed novel transient cell states in both the stem and differentiated cells that are specified. In the organism, this defines changes in gene expression frequency during something like regeneration or some other process. So, again, this is the ideal case. But in a lot of cases, you don't necessarily know what some fluctuation in RNA means. It's just kind of there's a lot of, noise at different scales. You have environmental stress. You have, you have to characterize what's going on in that cell type. So that's always the challenge there. But that's one tool. And then, of course, we have genome-wide epigenetic analyses, which are the analysis of, say, like methylation, which is this process that drives it mediates gene expression. You have these methyl groups that sit on top of certain parts of a gene, and they facilitate transcription of parts of the gene. So a methyl group will sit on top of different parts of a gene, and they'll either be opened or closed. And when they're open, they enable certain transcripts to be expressed, and when they're closed, they disable those transcripts to be expressed. And so there are they've been able to define different switches in genomes. So, like, for example, you do this sort of methylation analysis where you identify all these methyl groups, and then you look at their state, whether they're open or closed, and that can be mapped, say, to some phenotypic outcome, or it can be mapped to what the gene expression is doing. So, there are ways you can look at those. You can look at sort of a methylation analysis, bring it to bear on gene expression, and it tells you something about some of these, what they call epigenetic changes to the genome. So that's another tool that's useful. And then, so these are all useful sort of ultimately in telling us things about, development and differentiation and other sort of processes that involve cells, maybe cell populations, or single cells. Now, the problem with looking at these things in cell populations is that you're doing a lot of averaging and average behavior. So this is a little bit, this is another tricky aspect of it, is why, which is why it's, interesting, but it's, we're still kind of thinking about the implications of it. So these circumstances, combined with our discipline's diversity of experimental and intellectual approaches, offer unimaginable opportunities for developmental biologists, not only to discover new biology, but also to reveal entirely new principles of biology. So this, I don't know if there are any nice pictures in here, but this basically the idea here is that you have, this is an example of regeneration. This top panel is the flatworm. So this is a model organism that has all the cells in this flatworm are totipotent, meaning if you take any one cell and you put it in a new location, put it like they culture these worms in dishes. So if you take one cell out of this flatworm and you put it in a new dish, it can grow an entirely new flatworm. Furthermore, it retains a lot of its morphogenetic program, which means it can form this morphology that looks exactly the same as this one. And
okay, now they just need to add the physics.
Yeah. And so there's been work done on this in terms of bioelectricity. Like if you take one cell and you put it in a new context and you can grow a whole new individual, how does that happen? I mean, there has to be some sort of three-dimensional coordinate system here. But what is it? So
it's called an egg.
Yeah. So I think Mike Levin's done a lot of work on like bioelectricity and how that sets up this sort of spatial reference frame. But, again, this is a problem that isn't reducible to necessarily to gene expression and methylation and epigenetic modifications, at least in the way that we're measuring them. So they're, in this paper, they're kind of proposing that a lot of the tools that we have are molecular. And of course, there are other tools that we have. We have ways to measure physics. We have ways to measure the bioelectric state of things. So these are all things that will probably be useful for understanding the assembly of this phenotype, especially if you have something like a flatworm. So there's also then this organism here. This is just a fish with a fin, and this shows fin regeneration. So this is like, in this case, the head is developing from sort of an embryonic state. In this case, the fin has already been developed, but it's been damaged somehow, and so it's being regenerated. And so they're arguing is that the same and we know this, that similar processes are at work to rebuild the tail.
Note that cutting off the head doesn't work for a fish.
No, it doesn't. You can cut off the head of a flatworm and you can get a new head, but you can't do that with a fish. And the reasons for that having to do with like the way the the sort of the regenerative capacity of the organism. So some organisms
that's an excuse.
Yeah. But we really don't know. Yeah, we don't know the full extent of why these things are the way they are.
Yes, why can't we grow a new head? All those beheadings that were done during the French Revolution, they've grown back.
Yeah.
Any rate, Bradley, my answer to people who think that developmental biology is over is in the chat.
Okay. So let's see. Yeah. So Dick said that he has his paper, Are We on the Cusp of a New Paradigm for Biology? The Logic of Molecular Developmental Biology versus Janus-Faced Control of Embryogenesis via Differentiation Waves. Yeah, this is the paper that you did, I think, with...
last year.
Yeah, so this is...
I did a tutorial with Rob Stone.
Oh, yeah, Rob Stone, yeah.
That's a separate paper.
Oh, okay. So this is in Biosystems. Okay. Thank you, Harikrishna, for attending. So yeah, and so this paper that we have here is just talks about talks about plasticity in development. So plasticity, of course, is this other idea that you have an adult phenotype and you're actually able to regenerate it in a way similar to development. So this is, we're using sort of the same molecular pathways and some of the same mechanisms. And so this paper is very much focused on the molecular aspects of it. They don't really talk about the physics too much, which I think are, there's a lot of interesting stuff going on with physics, of course, we've talked about in the meetings. And we always have these nice figures of different things going on in the biology. They make these really colorful figures. So this one shows the relationship between transcriptional state and time. It's showing these fluctuations in transcriptional state and how that can affect sort of the progression of different developmental processes. So there's not only gene expression, but there's also this gene expression noise or fluctuation that is important in setting up a lot of this stuff and driving it forward. And then you have these type of graphs where you have RNA over versus time. So you have different tissues, and if the RNA for those that define those tissue types are expressed at certain points in time, you get the correct tissue. If you don't, then you get defects in the tissue. That's what they're showing in there. So that's that paper. And then there's this other paper called the Aerotechnic Organization of Developmental Biology. And so this kind of talks about, they're kind of talking about the presence or absence of theory in development. And of course we know that there's theory and development in this group, but some people, are debating, what the relative value of it is. So in this book chapter, Developmental Biology is the science of explaining how a variety of interacting processes generate the heterogeneous shape, size, and structural features of an organism as it develops from embryo to adult, or more generally through its life cycle. So although it is commonplace in philosophy to associate sciences of theories, such that the individuation of a science is dependent on the constituent theory or group of models. So this would be like where you associate a certain scientific field with certain theories or models. So, you would come up with, I think in physics, you would have certain models. If I said like quantum loop gravity, you would have like a series of models or theories associated with that. And, the same thing happens in a lot of other fields. It is uncommon to find presentations of developmental biology making reference to a theory or theories of development. For example, in the third edition of Essential Developmental Biology, 3 families of approaches are described, and those are developmental genetics, experimental embryology, and molecular and cell biology. So those are kind of like the sort of the consensus views of how to look at developmental biology. And that's kind of, experimental embryology might include like a vast umbrella of approaches, or that's an umbrella for a lot of different approaches. The same thing probably holds true for molecular and cell biology. There's a lot of stuff under that umbrella. So it's not really, that's kind of a family of approaches. And the appendix contains a catalog of key molecular components. So genes, transcription factor families, including factor family, cytoskeleton, cell adhesion, extracellular matrix components. However, no standard theory or group of models provides a theoretical scaffolding to the book, nor is any mentioned. So in that book, they didn't have any overarching theory. And so again, we know that we have theories of development, but the idea is that's not something that they, in the textbooks at least, they don't organize it by, this is a theory that unifies everything.
Yeah.
And so...
Hey, Bradley?
Yeah.
You're talking about this led to something that you might want to try. Look at the chat.
Regenerating networks. Okay.
Yes, it seems to me that just like was done with the vehicles thing.
Right.
One could start with networks and ask, what does it take a network to regenerate itself? We have examples in some of the Wolfram patterns.
Yeah.
Okay, because the Wolfram pattern can regenerate itself.
Yeah.
Okay?
Yeah.
So I'm suggesting that maybe some sort of general theory about what does it take for a network, even though it may not be a real thing. What does it take to make a network that can regenerate itself if you cut parts of it off?
Yeah.
I mean, it could be a branching network, or it might have some loops in it. But either way.
Right.
Okay. So, rules for regeneration of networks: and can they work, and can they reproduce the network?
Yeah. I'm just writing this down.
Has anyone done that?
I'm not sure. I've seen some papers where they kind of maybe go in that direction, but I don't think anyone's ever established it in terms of rules or in terms of. I know people do a lot of things with things called motifs. So they look at motifs of networks, but this is largely like usually something like gene expression. It could be like even like a connectome of neural cells.
I'm saying you actually have a network, cut part of it out, and see if it can grow back.
Okay, yeah. Now, I don't think anyone's done the grow back part.
Yeah, I think that'll be interesting, and it might be applicable to developmental biology. I don't know.
Yeah.
It would be a nice exercise, even if it isn't.
Right.
Okay? Might be able, if we can get anything done that way, it might even get into the cells issue.
Oh, yeah, it'd be great.
Okay?
Yeah.
Okay, have some bad dreams about it.
Yeah, well. Yeah. So, I guess, just to finish up this paper, the absence of any references to a theory of development is puzzling on its face. Why is it so difficult to identify or isolate a constituent set of models?
The absence is mostly because this guy didn't read the literature.
Yeah, I know.
That's okay.
These interpretations are possible. The first is a lack of reference to theories does not indicate the absence of a theory. One can reconstruct a theory or theories of development out of a relevant discourse. The second interpretation is a lack of reference to theories indicates an immaturity in the field. And then, that kind of goes into the... So that's what he'll argue, or the third option is what he'll argue for, which is that the lack of reference to theory should be taken at face value. So, I mean, that's...
that's nonsense. We can go at least back to Wolpert.
Yeah,
in 1969 with his positional information. That certainly was a theory.
Right.
It may or may not be right, but was definitely a theory.
Yeah.
So, the strong statement, there aren't any. Absence means he didn't read the literature.
Right.
Okay.
Yeah. So, I think it's like some food for thought. And I think, there's I don't want to say there's an aversion to theory by some people, but there's definitely...
in biology, there definitely is.
Yeah. But, it's always...
It's called absence of theory by not funding theorists.
Right.
Okay, it's very simple.
Yeah.
It's been around for decades.
All right, so the position they take in this paper is that there's a lack of reference to theories and that's just the state of developmental biology as a field. So the argument has two dimensions, one positive and one negative. The negative dimension consists of showing why the first two options, which are these up here, are not preferable in this context. On the one hand, claiming that developmental biology is immature is not a viable interpretation. So there's plenty of data, but the argument is that it's not very well linked to theory. It's not really a nascent science. We know a lot about embryos, but we don't necessarily know how they're integrated, at least theoretically. So this is his argument here, and I don't know. I guess it's more about like whether theory is going to be a guiding force in the field or not, rather than whether there are theories or not. So this is kind of a, this could be sort of a chance for wiggle room on this, but on the other hand, holding that developmental biology already has a theory or theories costumed in different guise, not referred to as such by developmental biologists, is a possible interpretation. So, we've talked about things like positional information or differentiation trees and differentiation waves. Those are of course theoretical, but are they something that we use in the field in general as an interpretive mechanism, or is it just something that we kind of deal with separately in certain cases? So this is kind of an interesting relationship between the two things. Let's see. So in his infamous book Against Method, Feyerabend, who was a famous philosopher of science, argued against the presumption that there are universal methodological rules that govern scientific inquiry. This author argues against maturity in the sense of a single conception of what it means for science to be mature that is applicable across all areas of scientific inquiry. The association of maturity with a science having a central organizing theory is a long-standing idea. So, the idea is that maybe not all sciences have a central theory by which everything is explained. Maybe, having multiple theories that explain certain aspects is something that is sort of the nature of developmental biology. This is, of course, as opposed to evolutionary biology, where natural selection is basically the mechanism, and Darwinian evolution is the consensus view. So that's basically, so when he says Aerotechnic in this paper, so he offers an account of how problems play the organizing and guiding role typically presumed to come from theories. So the idea of Aerotechnic, or pertaining to questioning, sort of frames how developmental biology is developed. So developmental biology is developed around questions and about, different problems that come about. So it's very different from like a field where you have a lot of sort of overarching questions that might be best explained by a theory from the data. And, it has sort of it flows naturally from that. In developmental biology, this is something that is, the explanatory framework is really revolves around different questions and aren't necessarily integrated. So this person explores a recent research paper in mammary gland morphogenesis to illustrate this argument. And so this is in the paper, down further in the paper, and they can talk about this. This is a figure here, the relative frequency of theories, theoretical, and article keywords from different domains of scientific inquiry. So this is the percentage of articles in this domain that have theory in the title. And as you can see, anatomy and biomechanics have 0% of articles devoted to theory, whereas economics have almost 100%, engineering has almost 100%, where the frequency of theory exists. I don't know how engineering was calculated, but in any case, you see that there's variation between different fields. And so I guess the point of this graph is to show kind of how closely theory is tied to empiricism. So when you have a paper, do they reference theory or not? And so, in a lot of engineering papers, for example, have a set of equations that they review, and so that's often implicitly theory. And so they mention theory in the paper. There's an engagement with theory, there's a dialogue, and in other fields there really isn't that. Development is here, actually, and that's 0%, just like anatomy and biomechanics. And that's maybe an artifact of people not really engaging with sort of mathematical models or other types of theories that may exist. And so that there are multiple explanations for this. So again, textbook presentations of developmental biology, it's aerotechnically organized, so a lot of textbooks are organized around questions, around some of these themes, but not necessarily around theories. And so this kind of goes through the textbook structure for this book, Essential Developmental Biology, which there are two versions, the second and third edition. So they kind of lays out the changes in these two editions to show that basically the sort of presenting the field to a bunch of students, you have to lay the groundwork, but the groundwork is generally these topical areas and not necessarily necessarily theoretical areas. So they're approaches to, say, developmental genetics or experimental embryology. And they typically lay out maybe experiments or techniques, and they're not based around theory. Major model organisms, again, these are model organisms where we know the biology, but there's no real theory around them, and certainly no theory linking all of them together. Organogenesis, again, these are examples, and then growth, regeneration, and evolution, again, examples. And they kind of get into evolution and development at the end, which is not, it's kind of, because it's at the end, it tells you sort of the priority there of where it fits. And I think, this is interesting. If you're familiar with Evo-Devo, which is a theory of evolution of development, one criticism of Evo-Devo is that it's not really a theory, that it's just a collection of neat findings. They package it in this very superficial explanatory framework, and then they present it as sort of an alternative to the evolutionary synthesis. And of course, it's not really a theory in the conventional sense. It doesn't predict necessarily. It may predict things, but it doesn't really put them into a larger framework. And so this is a common, it's a common criticism of Evo-Devo, and it's also sort of a criticism, I guess, of this approach to development where you're laying out the facts, you're laying out the models, but they're model organisms, so they don't really have any integrative power. Just examples that you're picking out. I think this is, so more analytically, the space of problems in the field of development is characterized in terms of 5 variables: abstraction, variety, connectivity, temporality, and spatial composition. And so this is something that the way we kind of view development and some of the different ways that, you can look at development. The values given to these variable structures, a constellation of research questions within the broad problem agendas corresponding to generally delineating phenomena. So you could imagine, if you want to really connect development or theory to developmental biology, you've got to focus on these 5 different variables and kind of bring the theory to bear on each of these. I think really to get people's attention. Then Scott Gilbert put out a textbook, the 10th edition of Developmental Biology, and a consistent feature or multiple editions of this book is that developmental biology is constituted by a set of core questions. So you have these different questions that the book is organized around. The first being the question of differentiation. So how can a single cell generate so many different cell types? The question of morphogenesis. How can the cells in our body organize themselves into functional structures? The question of growth. How our cells know when to stop dividing? How is cell division so tightly regulated? And so on and so forth. And so it gives us, it's organized around questions rather than like this conforms to this prediction of this theory. So that's what he means by aerotechnic. And this again is different types of morphogenesis. So you can see that there are all sorts of different types of morphogenesis. And really this brings up another point, which is that you're dealing with very diverse systems. So, the question isn't that there isn't a theory, but that the theory has to be very broad to explain all these different modes of variation, or all these variants of a common set of processes. So this is this is morphogenesis. And of course, we know that you have reaction diffusion morphogenesis as a theoretical model. You have positional information as a theoretical model. But then if you try to put this in the context of all these different things that you're observing in nature, is there one theory that can bridge all of these? And the question is probably not yet, but we have candidates. And of course, there's no reason why you couldn't apply theories to each of these different cases. Like, for example, if you had reaction diffusion, which one of these examples would it explain best, and which ones would it fail at? And so, this is maybe a road to another paper where you could say, well, there are all these different modes of development, and we have these different candidate theories. Which ones fit which context the best, and which ones fail? And so, you have this sort of mosaic of theories that might be useful, but, breaking that down for people, especially practitioners in the field, which theories are best for which modes of development and so forth, which examples. Because a lot of developmental biology is driven by examples that you might see under a microscope. And that makes sense because if you're actually engaging in the field and you have a lot of variation, you might want to know, my model system, how do I explain what's going on there? How do I explain what's going on in another model system? If you're comparing model systems, however, it becomes hard to really understand that thing is a common process. For example, we've worked with C. elegans and zebrafish and xenopus and axolotl in this group. We've worked on these different model organisms, but they have very vastly different developmental periods, what the developmental period looks like in terms of its mode. And so, is there a theory that can bridge all of those? We've argued in past papers that you can find theories that bridge those. But it's controversial, I would say, and it's very hard to really kind of make the case very deeply that these theories are connecting all of these things. So this is this paper is nice because it compares some things on the ground like textbooks and how they're approaching the field and how they're kind of framing the thing, how they're framing the material in the field for people to understand its structure. And so it doesn't, there isn't a lot of theory that gets introduced. I mean, usually in a evolutionary biology course, you would learn about evolutionary theory. You would have examples of things in nature. You have examples in evolutionary theory, like the peppered moth or other models, like simple Hardy-Weinberg equations, things like that. But you don't really have to this level of sort of question-driven presentation where you have all these different subtopics and then you're asking questions, but you're not really integrating them very much. Okay. So this it kind of goes through this question of sort of how the heterogeneity of developmental biology is kind of driving this sort of question-driven approach. So this is really kind of a key thing, and I think it's, maybe that's something that needs to be overcome. Maybe there needs to be a developmental biology textbook that doesn't focus on the questions so much, but maybe focuses on theories. Or maybe we need to do a session where we kind of work out these theories, or a series of sessions where we kind of work out these how theories connect to a lot of what's going on in developmental biology. In fact, that's why I do a lot of these paper presentations, because I like to bring up these topics and I like to see how we can maybe fit theory to these topics or unpack them in a similar way. So make connections with other parts of development. This is a sort of a thing I've learned from this paper, is that you can have that we have to understand the structure of the field that we're engaging with in order to really get an appreciation for why theory building is so hard.
Yeah, the first time I applied for a grant when I became a professor, very first time, they said, "What does a theoretician need money for?"
Yeah.
And I got turned down.
Yeah.
On that basis. Or they gave me a small fraction of what they asked for.
I've noticed that even I forget the guy's name. Anyway, they'll say something, and it looks like they're making a statement that this is true, and then they'll go, "Oh, well, maybe I'm not correct." And they say that in biology all the time. And as an engineer, you take the paper and you go shred, shred, shred. No information.
Yeah.
It makes it have no information, and yet they've positively stated their theory and their conclusions.
But this is the Alice in Wonderland approach. I can swallow 3 contradictions for breakfast every day.
Okay. Well, it's very frustrating because you can quote this person as saying this, but and then they say the opposite, and you go, they sort of said this.
Yeah.
And it's like, and then you get an engineering professor reading what you've written, And they go, well, why did you say they sort of said this? Like, that's not a proper statement.
Because they spoke out of both sides of their mouth.
I was like, wow. Well, this makes it difficult for me some days. Anyway, yep.
Yeah. Okay.
I guess I could turn this on. I don't know. I am getting fiber optic. It's been hooked up to my house, but it hasn't been attached. So I asked when that might happen. They said September. So I don't know. I have fiber optic, but it's not hooked up to anything except the
Make your
house.
own.
Yeah.
I'm made in China now.
Yeah. Go get a Huawei receiver.
Yeah. Right.
Oh well.
Anyway, Bradley, I can think of examples of regenerating network right off the bat.
Okay.
Suppose you had a stochastic simple branching process.
Yeah.
Okay, but the probability of generating a new branch kept decreasing, so that the thing essentially reaches the terminal size. It's analogous to a series which converges.
Okay. Yeah.
Okay. Even though the number of terms is infinite, it does converge because the steps get smaller and smaller.
Yeah.
Okay, so I think we could actually construct regenerating networks.
Yeah. Yeah.
They'd be simple. Start with very simple ones like that.
Yeah.
Okay.
Yeah, that would be great. I'll look
Okay,
into
and
that.
they're easy to draw because they're just a bunch of lines.
Right, right. You just have like a branching process. Yeah.
Okay.
All right.
But that one would be self-limiting. Okay.
Right.
And size is an important part of developmental biology, so that's the way to do it. One way to do it.
Yeah.
Okay.
Yeah.
Would that get into why salamanders have such large genomes? Is because they have a complicated life cycle, or
I have no idea. Because they used to be smarter than people.
Oh, okay.
They got on Earth well before. Remember, they're very close to the first tetrapods.
Right.
Okay.
So they were the first smart fish, smart enough to walk on land. I don't know if that has anything to do with genome size. But
Yeah.
anyway, I think we can think of a bunch of simple examples of regenerating or self-limiting networks.
All right. Yeah.
Okay? And cutting is obviously easy.
Yeah. Yeah.
You just cut out a part of it. That's all. You get a simpler network, but you have growth rules.
Yeah.
If the growth rules regenerated or limited, then you have a regenerating network. Okay, so I think it's a neat possibility.
Yeah.
I've never given much thought to regeneration,
Yeah.
but I think by simplifying it down to a simple network, we might be able to do it.
Right. Yeah. It's like some sort of what they call sort of a toy network where you just kind of generate a.
Yeah, and it would fit in with the idea of the snail shell patterns regenerating themselves after the shell's been broken, because they got to start over again.
Yeah.
Okay, so since they obviously do start over and reproduce the same pattern, they if they really have a network basis, then maybe we've got a simple model there.
Yeah. Yeah. Sounds good.
Okay.
Yeah.
Okay, good. If I got you excited, let's do a paper together.
Yeah. Yeah.
Okay.
All
We
right.
won't ask you to join because you'll get in more trouble.
Yeah. Yeah.
I'll get into trouble, and I'm in networks. I'm not up to date on any type of
Bradley is. That's one of the
Yeah.
tremendous advantages.
Yeah.
Okay.
Yeah.
I'll take pictures of gumballs or something, and and I'll get enough trouble over that. So, and I'll just say, but I had the machine. I just put a little thing in it, and I took pictures for somebody. Yeah, he's probably not going to buy it, but you know.
Yeah.
Oh, Susan, do you know what a tektite is?
Yeah.
All right.
Okay.
That was a good meeting. All right.
Okay.
Yeah.
Okay. Jot down some ideas on regenerating networks.
Sounds good. Yeah. Talk to you next
Okay.
week. All right.
Okay.
Thanks.
Bye.
Bye.
Bye.